Cheers. Thanks a lot, Tom. You're marvellous. I'll start. So, disease mechanisms in viral encephalitis. So as we've heard from Nick, the most common cause of viral encephalitis is HSV type 1. And really, adjunctive anti-inflammatory therapy, which limits all the inflammation that we've been hearing about without impairing clearance of the virus, is desperately needed, because many of these patients, despite acyclovir, will die. And as we've heard, the majority of survivors are left with significant neurological or cognitive sequelae. During my PhD, I did some work looking at CSF and serum samples of patients, looking for chemokines and cytokines, and identified that across the board, the balance between the interleukin-1 pro-inflammatory family relative to its endogenous antagonists, the closest correlates of cerebral edema and poor GCS and poor outcome at discharge. But really, we need a mechanistic understanding of how what is predominantly a neuronal infection leads to inflammatory mediated cascades, blood-brain barrier breakdown, leukocyte migration into the brain. And understanding this is critical if we're ever going to identify targets for targeted therapy. And clearly, understanding this for HSV encephalitis has translational implications for encephalitis more broadly. Clearly, in a human, you often get one lumbar puncture or two if you're lucky, so it's a challenge to study it. So over the last few years, I've been developing an intrathecal mouse model where mice are inoculated through the not yet ossified sagittal suture, either with HSV or with sham infection with PBS. And then mice can be treated daily peritoneally with acyclovir treatment or sham treatment with PBS. And we can assess the morbidity using the Thomas encephalitis score. And I've shown using this model that treatment with acyclovir does expedite the clearance of replicating virus. And by day 4 and day 5, replicating virus is largely cleared from the brain, although, as we've heard, clearly viral RNA will be present. So I wanted to evaluate whether this was a useful model, and I've shown that via this route of inoculation, there is a predilection of HSV to the campus, whether we stain with a HSV-labeled FITC or use a GFP-labeled HSV. In addition, infection with HSV leads to leakage of an intravascular Evans blue dye into the brain, and also leakage of a FITC-labeled albumin, both around the meninges and also within the cerebral cortex. And we can quantify this. And in this severe infection model, despite treatment with acyclovir, morbidity and mortality remain not largely improved. So I wanted to understand what might be happening in these brains in response to HSV. What are the leukocyte populations? And identified that as early as 6 hours following infection, but definitely by 72 and 120 hours after infection, there's this huge predominance of neutrophils and to some extent inflammatory monocytes also into the brain. And there is no significant difference in reducing the levels of these leukocyte populations in the brain in those mice that have received acyclovir. So I then looked at whole brain Luminex to see which were the cytokines and chemokines that might be pertinent to this. And the two most significantly upregulated that remained upregulated despite acyclovir that reflect neutrophil and monocyte migration are CCL2 and CXCL1. So I wanted to understand if there was a phenotypic difference if we were to abrogate the signaling of these mediators. So using CCR2 knockout mice in red, which lack the cognate receptor for CCL2, there was actually an increase in cumulative morbidity. And conversely, in CXCR2 knockout mice, which lack the receptor for CXCL1, there was a marked reduction in terms of morbidity. So I wanted to determine really what these knockouts meant, if there's a phenotypic difference, what's happening within the brain itself. And demonstrated that in the CXCR2 knockout mice, there was a marked reduction in neutrophil migration, particularly at day 3, and in the CCR2 knockout mice, a marked reduction in monocytes. So they are playing a role in migration. What does that mean for viral control and for the integrity of the blood-brain barrier? Well, there was no significant difference between wild types and CXCR2s, but the CCR2 knockout mice lacking monocytes had increased levels of propagating virus within their brain, despite treatment with acyclovir. And the blood-brain barrier integrity, as measured by the brain to serum albumin ratio, was reduced in the CXCR2 knockout mice. So it looks that CXCR2 might have a role for neutrophil migration, and it might have a role perhaps in what's happening at the level of the blood-brain barrier. So if CXCL1 was of interest, the next thing was to see, well, what are the cell populations within the brain that are expressing this? And this is the CXCL1 isotype control. And we compare that to HSV infection, there was evidence that it was the GFAP positive perivascular astrocytes and also to some extent neurons, where there was co-localization of CXCL1 staining, suggesting that perhaps these are the neuroglial populations producing this mediator. To really thoroughly evaluate this, we repeated the experiments but conducted them in vivo. So here we inoculated murine neurons, microglia, and astrocytes with cumulative doses or inoculums of HSV, and found a dose-response relationship in terms of the production of both CXCL1 and CCL2 by astrocytes. But of course, this flies against the conventional wisdom that, at least in viral encephalitis, that microglia are this neuroglial population, the predominant producers of inflammatory mediators. So we looked at what was produced in response to infection and found that indeed microglia did produce members of the interleukin-1 family in response to HSV infection, which then made us think perhaps there might be a paracrine response between microglia and astrocytes. So what we did then was it incubated neurons, astrocytes, and microglia with two different doses of interleukin-1 and demonstrated that both neurons and particularly astrocytes upregulated their expression of CXCL1 when incubated with IL-1. And then we can actually block this effect completely with a co-incubation with the interleukin-1 receptor antagonist. And then I really wanted to study if CXCL1 has a role in neutrophil migration, where is that role? What is its role in this process of neutrophil migration? And these are intravital microscopy studies where I perform microscopic cranial windowing surgery on mice and replace portions of the skull with glass. And it allows us to use multiphoton lasers to make fluorescent cells within the brain glow. And here you can see 6 to 8 hours post-infection in the wild-type sham-infected mouse, these green cells traveling down the lumen of the post-capillary venule in health. And what a video I'm showing, all of these will be lysem GFP positive mice. So in the context of this model and this time point, these are neutrophils. But then in response to HSV infection, you can see this marked migration of neutrophils into the brain and this perivascular swarming of neutrophils within the cerebral cortex. Then repeated the experiments, but in the CXCR2 knockout mice, and identified quite strikingly that although neutrophils were called to the cerebral vasculature, they were by and large, for the vast majority, unable to migrate into the brain whatsoever. But then to determine if it really was CXCL1 that was the key CXCR2 ligand, I repeated the experiment, but now in a wild-type mouse, treated 30 minutes prior to imaging with an anti-CXCL1 antibody, and showed that actually CXCL1 had a non-redundant role, not in rolling, arrest, or crawling, because the neutrophils in the intraluminal accumulation were still able to undergo all of those steps of diapedesis, but they were unable to undergo transendothelial migration. So I wanted to see whether there was something about these different phases of migration that might be responsible for blood-brain barrier breakdown. So what we can do actually is, the reason you can see that blood vessel is because the mouse has received an intravenous Q tracker, which is bound to a protein which should remain within the blood vessel. And we can compute the vessel surface and actually calculate the volume of that intravenous tracker, which is within the blood vessel over the course of time. And you can see that in wild-type mice in response to HSV infection, there's an accumulation, even early in infection, of red Q tracker outside of the blood vessel. And then this is completely abrogated in CXCR2 knockout and wild-type mice treated with anti-CXCL1. And this markedly statistically significant findings. So the next question was, is it really transendothelial migration? Is it something about that step of the process that's really driving blood-brain barrier breakdown? So I went on to quantify each of the stages of migration relative to the breakdown in the blood-brain barrier shown in red. And it was actually only the abluminal accumulation of neutrophils that correlated with breakdown in the blood-brain barrier. And it's clearly much, much more work to do. But as the video on the right demonstrates, you can see here a neutrophil which has already undergone transendothelial migration and is accumulating within that perivascular space. And periodically, you'll see a splurge, for want of a better word, of red out of the blood vessel as this neutrophil effectively is punching holes in the endothelium. And the questions around are these reactive oxygen species? Is it netosis? And lots of work that's ongoing now through an MRC-funded study. So the conclusion of this research really was that HSV seems to be able to replicate in neurons. It seems to produce damage and pathogen-associated molecular patterns, which can activate microglia to produce interleukin-1, which can stimulate astrocytes to produce CXCL1, and also neurons to produce CXCL1. But that also pathogen-associated molecular patterns on their own are sufficient for astrocytes to produce CXCL1, and that CXCL1 does appear, at least in this model, to be the critical ligand responsible for transendothelial migration of neutrophils into the brain, and that it is this process in this model which is most closely associated with cerebral edema. So the question, of course, with all of this mouse work is really, what does this mean for the patient in front of us? Just got MRC funding for a study to do exactly this sort of work. Working with Tom, who's, as you've heard, is leading the DEX-NCEF study. We'll have 80 to 90 patients who have been randomized either to dexamethasone or placebo as an adjunct to acyclovir. What we'll be doing is we're quantifying the volume of cytotoxic and vasogenic edema on neuroimaging, comparing that to the leakage of albumin from the serum to the CSF, and then using targeted and whole proteome techniques to identify in patients what are the mediators most closely associated with vasogenic edema, which is that due to breakdown of the blood-brain barrier, whereas we think cytotoxic edema predominantly reflects a viral cytopathy. Then armed by this clinical data, we'll go back to the mouse model that I've established, where we will then abrogate the signaling in a combination of knockout mice and also mice receiving neutralizing antibodies, assess the morbidity by the Thomas score, assess the leukocyte populations in the brain by flow cytometry, assess the cytokines and chemokine expression as a consequence by Luminex, and then perform single-cell RNA sequencing to demonstrate where along the pathway there are associations between infected and uninfected neurons and other glial cells and the inflammatory mediators of interest. And then these findings will be taken forward in a blood-brain barrier model so that we can really evaluate if the hypotheses generated by single-cell sequencing are held up in vitro. So in conclusion, viral encephalitis is a devastating disease, major components of which may well be host-related, targeted therapy which mitigates the detrimental inflammatory response, but without abrogating parts of the inflammatory response which are critical for viral control are desperately needed and may well be possible. And of course have translational implications for the many causes of viral infection we've heard about from Nick, for whom we don't have effective antiviral therapy. And there may be aspects of the host immune response which are absolutely critical. But if we're going to do this, I think we really need a network understanding of the neuron and glial interactions which lead to the downstream mediators of interest, if we're ultimately to develop therapies which improve patient outcomes. Thanks to the postdocs who did some of this work, PhD fellows and clinical fellows and others. Thanks to all the NeuroPaces, NeuroAccess guys. Thanks to the COVID CNS study team. And I've literally, as I was listening to Nick's call, just had the text message to say that we've recruited our first patient. So that's incredibly exciting. And I'd be delighted to invite interest from those in the audience if you'd like to email me later on about the COVID clinical neuroscience study, where we're really delving into similarly trying to understand the inflammatory response in patients who've had COVID-related brain complications. Finally, thanks to Brain for the invitation, for Tom for sharing, and for you all for listening.
Thank you, Ben. Brilliant. Fantastic. Thank you very much. So please post your questions now using the Ask a Question at the bottom. There's a question from Dr. Saikit Dar. Can you please comment on the susceptibility and clinical severity of HSV encephalitis in immunosuppressed patients, in particular with respect to the innate immunity?
Yeah, thank you. So in clinical terms, I think we tend to think that sometimes patients who are immunocompromised will present with HSV encephalitis in a more unusual way, perhaps more subacutely, and perhaps because they don't have this profound inflammatory response that actually leads to raised ICP and seizures and the other things that cause depressed GTS and lead to hospitalization. And the other thing is some of the temporal lobe presentations of HSV encephalitis are more common in immunocompromised patients, such as rhombencephalitis or cereballitis. In terms of the innate immune response, we know that TLR2, 3, and 9 are critical for the ability to identify replicating virus and those pathogen-associated molecular patterns. And in fact, patients that have mutations, particularly in, well, HSV is relatively rare, isn't it? So in patients that have a whole family that have all had HSV encephalitis, we can find that there are mutations in those toll-like receptors. The one that's been of greatest interest is the UNC93B mutation.
Ben, maybe I can ask, this CXCL1, I think you've demonstrated really nicely the role it plays in neutrophils migrating across the endothelium in the vascular endothelium in the brain. Do you think this is also the same in other vascular endothelium in other parts of the body, or is this specific to the vascular endothelium in the brain?
Yeah, thanks. It's an important question. People have done other studies where they've looked at non-alcoholic fatty liver disease, and they've looked at infarction, and they've looked at chemokines and neutrophil migration in other organs. And actually, it's predominantly CXCL2 which dominates the literature. So it's interesting that CXCL1 seems to have this potentially important role in the brain, at least in this model.
Great. Okay. Any other questions from anyone else? If not, I'll just ask one more quickly. I mean, the focus here is on neutrophils, whereas in patients we mostly see lymphocytic CSF. Presumably that relates to the timing and the fact that patients come to hospital early, does it?
That's right. Yeah. So I think
patients come late. Sorry. Yeah.
Patients come late. Yeah. So yes. Yeah. I mean, as you know, neutrophils predominate early in disease and lymphocytes later. But interestingly, when you look at post-mortem studies further down the line, actually the hemorrhage and necrosis further down the line leads to a secondary wave of neutrophil migration. So I think our model is one of early and severe disease.
Great. Okay. Fantastic. Thank you very much.